Good morning, everyone, and welcome to the webinar. This is Rebecca Baillie, and today we're going to do things just a little different. So today I will be acting as both moderator and a contributor to the webinar. Before we jump into the webinar, I wanted to let everyone know that you can ask questions at any time during the webinar by entering the questions into the questions pane on your GoToWebinar side panel. So here with me today are Christina Friedrich and Jake Nichols, who will also be part of this webinar. Christina is the Chief Engineer at Rosa & Co. She brings over 20 years of experience in applying mathematical models of biologic systems to overcome development challenges faced by pharmaceutical and consumer product companies. Christina has spearheaded numerous modeling methodologies and contributed to foundational patents, including on the use of virtual patients and populations to explore biological variability and uncertainty. Jake is Director of Medical Affairs at US WorldMeds. Jake is a pharmacist in a long-term recovery from substance use disorder who has been working in the addiction field for over 12 years. He served in various roles throughout the pharmaceutical industry, including clinical operations, education, and consulting. He also regularly facilitates parent and patient support groups for those struggling with substance abuse, and we are so pleased to have Jake with us today. So today, we are going to talk about an instance in drug development when it was not possible to run clinical trials to show efficacy. So Christina, if you'd like to show the slides.
One moment. Are you seeing my slide? Okay.
Yes.
Okay. And I will turn my camera off just to preserve bandwidth here. Okay, go ahead, Rebecca.
So if you can, yes. So today we will be talking about an instance in drug development where clinical trials were not necessarily the solution and were not possible to do. And this is in regards to the current drug overdose epidemic and the use of a new dose of naloxone to reverse opioid overdose in that situation, in the overdose victims. So we're going to talk a bit about a model, and this is one of those instances where pharmacological modeling can help to supplement a regulatory submission. And we're going to focus on fentanyl and naloxone. And I'd like to open the floor at this time for a moment for Christina and then Jake to give a brief statement on this before we go into the rest of the webinar.
Yes, thanks, Rebecca. So I think just from a modeling perspective, this is a very interesting scenario. As I'm sure many people know, there is a drug epidemic. The synthetic opioids such as fentanyl are really taking over and causing a lot of harm. And naloxone is a drug that can be used to reverse the overdose. And the question was really with the the shift in the what's being seen in the field in terms of the use of synthetic opioids. Can we make an argument for a higher dose? So from a modeling perspective of naloxone to reverse the overdoses. So from a modeling perspective, is a very interesting question. I'll turn it over to Jake for a moment to just comment on, what makes this kind of development tricky in terms of demonstrating efficacy.
Yeah, from a clinical and a field perspective, I mean, this was in many ways an unprecedented move by the FDA to allow us to be able to present a model to really hinge the approval of the product on. And I think a lot of that was based on the fact that we had a medication which had been initially approved back in 1971 for the treatment of opioid overdoses, which we had really established a firm understanding of the pharmacokinetics and the pharmacodynamics of the drug itself. In combination, as was mentioned, with the truly unprecedented public health crisis and this questionable landscape of fentanyl analogs, this modeling was really pivotal in allowing us to get a higher dose naloxone product to market, and to be able to get it out there to people that truly need it. So it was extremely valuable. And again, I think we'll see more of this. There's been more models released around naloxone utilization, and it's gaining a lot of credibility in the field as well.
Thank you, Christina and Jake. And Christina, if we could have the next slide, please. So on the agenda today, we're going to discuss very briefly the background on fentanyl and naloxone. We'll talk a bit more about the opioid epidemic, and then we're going to go into modeling the mu receptor dynamics, some of the results and conclusions that were gained, and then talk a bit about how the modeling insights and how they were used. So Christina, next slide, please. Neither fentanyl nor naloxone are new drugs. Both were originally synthesized in the early 1960s, and both received approval from the FDA in the early 1970s. Fentanyl was originally approved for anesthesia and then pain, and it was originally approved as an IV drug. Transdermal patches for pain came out in 1990, and by 1994, there were warnings about abuse of fentanyl and these transdermal patches. 2013 marks the beginning of the current epidemic, and this is very specific to synthetic opioids. And in fact, it's known that opioids contributed to more than half of the overdose deaths in 2021, and that is expected to continue for when the data for 2022 and 2023 are collected. Naloxone was originally approved to reverse opioid overdose, and in fact, that's what it has been used for the past 60 years. In the mid-1990s, they started piloting what they call take-home kits. These are community kits that can be used by people who are not in the hospital, so first responders and community individuals who can administer naloxone and treat an overdose until medical personnel can arrive. In 2015, nasal spray naloxone was approved, and now the community kits are commonplace. So they are used a lot by individuals who are in key points within the community to try and reverse overdose. There is a large body of literature for the pharmacology for both fentanyl and naloxone. Again, they've been around both drugs have been around for 63 years, and there is a strong body of evidence to understand how they work and the PK for both drugs. Christina, next slide. The United States has a current epidemic for opioids. And since the pandemic in 2020, this epidemic has gotten considerably worse. The number of opioid overdose deaths has increased dramatically in the last few years. And even though during the first 9 months of 2022, the number of overdose deaths declined slightly from the prior year, they were still about 50% higher than the pre-pandemic levels. And those pre-pandemic levels are considerably higher than prior years and decades. The CDC has attributed the largest increase, or the increase in deaths, the overdose deaths, to illicitly manufactured synthetic opioids. And this would be fentanyl and fentanyl derivatives. And these opioids are highly potent. And next slide, Christina, please. Before 2013, there really weren't many deaths that were attributed to these potent synthetic opioids, but the number of deaths has increased radically over the last 10 years. And it now far exceeds the deaths due to more commonly prescribed opioids or even heroin and other natural derivatives. So one of the problems that is encountered is that these are often illicitly manufactured. They are different derivatives that may be stronger, more potent, tighter binding to the mu receptor. And because of that, there tends to be a really higher death rate from these opioids. Next slide, please, Christina. With the potent effect of these synthetic opioids, there has also been information in the published literature that resuscitations have required multiple doses of naloxone to reverse the overdoses. So this would be more doses at the 2 milligram IM or 4 milligram IN treatment levels. And this is probably, in fact, because of these synthetic opioids and the strength of these synthetic opioids. With that higher resuscitation notes, one of the things that was proposed was a higher dose of naloxone, and Zimi, a 5 milligram naloxone IM injector, was going to the FDA for approval. And with this naloxone, as both Jake and Christina have mentioned, clinical trials were just not possible. So Christina, if you can go to the next slide, how do you handle showing utility of a new dose if you can't do a clinical trial, and you particularly cannot do a clinical trial in the subject group that most needs it? And this is where mathematical modeling comes to the rescue. And you know, in this presentation, we're going to talk a bit about modeling the opioid mu receptor and its occupancy with fentanyl. We'll give different doses of naloxone, and then we'll investigate the hypothesis that higher doses of naloxone are beneficial. So let's take a quick look at the model. This is the model, and the model does include the PK of both fentanyl and naloxone, and the concentrations in both the plasma and the brain. The key part of this model is the competitive receptor binding. The model contains the mu receptor. It has receptor binding, internalization, and recycling of the receptor. And the competitive binding depends on both the ligand concentration and the K-on and K-off for each of the compounds. So this is a key feature of this model. And with that, I'm going to hand over to Christina so she can talk more about the model development and the model use.
Yeah, thank you, Rebecca. So as we've already alluded to, there are a number of challenges when you think about modeling in this scenario. So let's review some of those, and we'll tell you about what assumptions we took to move ahead with the modeling. So first of all, as is obvious by now, there's minimal clinical trial data available due to the ethical considerations that we already mentioned. So that's the whole reason why we are utilizing modeling to support the dose and receptor binding data. But it also means that there's, not a lot of direct controlled environment type of data available to inform the model. We also have to think about what are we actually simulating, because there's really a wide variety of different routes of administration. And we really this was not a controlled trial, so we did not know when fentanyl was taken. Was it intravenous, intranasal, transdermal, et cetera? There are many, many ways to take the drug. So we had to make some decisions about what we're actually modeling. So we wanted the model to be, as broadly applicable as possible. So what we decided was that rather than try to focus on a particular route of administration, we would just focus on the actual concentrations of fentanyl in blood and by extension in the brain. So we are starting our simulations at different blood concentrations of fentanyl, inferring the brain concentration and the receptor occupancy from that. And we are also assuming that we're basically starting sort of at the that certain concentration, and there's no additional fentanyl that is coming into the system or being absorbed after that time, which seems reasonable given that the subject has overdosed at this point. We also don't know when exactly the fentanyl was taken or how soon after fentanyl administration the naloxone would be given. So again, we're sort of getting around that complication by starting our simulations at that specific fentanyl blood concentration. And we're just going to assume that this is the time at which naloxone was given. So the naloxone meets the fentanyl when fentanyl is at a certain concentration and going down from there. And we're just going to focus on what happens after the naloxone is given in terms of displacing the fentanyl from the mu receptor. And naloxone is also going to be administered, as we already mentioned, by community or first responder personnel. So again, there's no particular dosing protocol that we can assume was followed. In the field, it is acceptable to redose if the victim is not responding. However, for the purposes of our simulations, we are focusing on a single dose of naloxone. we had to make some additional assumptions. As you might imagine, some of this can be a bit tricky. As Rebecca already mentioned, there are a lot of papers out for naloxone and fentanyl. We sourced a wide range of binding affinities from different papers. So we were quite thorough in reviewing all the reported numbers and essentially used a median value that seemed reasonable given the various differences in the way the affinities were measured, et cetera. Brain PK is always difficult to determine. We had to infer the brain PK based on various relevant data. I will show you a little bit of that in this presentation today. But again, what's important is, what you can measure is what's in the plasma. What we need to infer was how much is getting into the brain and how much of the mu receptor is actually being occupied by, when you see that level of the drug in the plasma. And we also had to, we did have data on the binding affinity, and we did importantly also have some data on the clinical effects from the field, and also data from some preclinical models looking at the how long the effects last. We did look into the specific transport kinetics for naloxone and fentanyl. They're quite difficult to confidently model in a mechanistic fashion, so we had to make some simplifying assumptions about the partitioning into the brain. And then we also implemented for fentanyl in particular some brain-specific reservoir. It gets quite complex, but there seem to be some inconsistencies between the clinical effects that continue to be seen when the plasma concentration is already dropping. So we had to make some additional assumptions around fentanyl's presence in the brain. Finally, there's actually quite a lot of variability in terms of the mu receptor dynamics from patient to patient. And in particular, the chronic opioid use, as you might imagine, actually has an impact on the mu receptor dynamics. So for the purposes of modeling, we decided to set those dynamic parameters, assuming that we were dealing with a chronic opioid user who would have delayed receptor recycling and some other specific properties. Okay, so as we mentioned, one interesting aspect of this is that there are different routes of administration for the fentanyl. You can kind of see that illustrated here. And let me just zoom in on this top part for a moment. So here we're looking at plasma concentration of fentanyl over time, following different routes of administration from IV, nasal, tablets, and this kind of lollipop that the patient can suck on. And what this shows is that, clearly you'll have different absorption rates and different Cmaxes in plasma. But generally speaking, you would, we're looking at a pretty consistent half-life. And so this seemed to be, confirm that our assumption of just, or our simplifying modeling setup of just starting with a particular concentration and then tracking the clearance of fentanyl is a reasonable approach regardless of the original route of administration. And you can see that, of course, the different administrations, again, are associated with different Cmax levels. Now in this slide, we are actually translating that into the fentanyl, or we're showing you the modeling results of the fentanyl in plasma in nanograms per mil. And what we're showing here is the plasma half-life. So the drug is decaying from, in this case, we're modeling a 50 nanograms per mil a peak concentration of fentanyl. And again, let me zoom in here for a moment. And that 50 nanograms per mil is a sort of a mid-range of the concentration that used to be seen in drug overdoses. As Rebecca already explained, there's, with the increase in more and more potent drugs and more fentanyl flooding the market, this is probably going up. These data are a few years old, but this used to be, a reasonable assumption for how much fentanyl might be in the system of an overdose victim. Somewhere below that is the estimated loss of consciousness range of fentanyl concentration. That becomes important in just thinking about how much of the fentanyl actually needs to be displaced from the mu receptor in order for the subject to regain consciousness. And then somewhere way down here are the actual levels that are used in the clinical setting for surgery, et cetera. And those same data are summarized again over here, just to give you a sense of the levels that we were able to find in the literature. Okay. This is just showing a couple of very important validation points. So as we discussed, there's the leap that we had to make from the plasma concentrations that we have data for to the brain concentrations, and then to the mu receptor occupancy given that concentration in the brain. So that's the way the model works, right? The drug is given, it shows up in plasma, it shows up in brain, and then it occupies the receptor. We were lucky to have some very nice data in the naloxone case that actually tied the peak plasma concentration to the mu receptor occupancy, which the model was able to reconstruct very nicely. And then on the fentanyl side, we were able to infer the that same relationship between the blood concentration and the receptor occupancy, given the various lines of evidence here from the literature. And what we're showing over here is an example where sorry, let me zoom in on that again. Okay, so here we're talking about the fentanyl receptor occupancy over 12 hours. So that 50 nanograms per mil, again, that was the concentration that was determined to be representative of an overdose probably a few years back. That's roughly equivalent to a 10 milligram transdermal patch. And you would expect actually that at that 50 nanograms per mil, you would get close to 100% receptor occupancy. As you go down from there, occupancy goes down. One other thing to note is that, of course, the greater the fentanyl concentration, the longer it takes to clear from the system. So you also have increased duration of the fentanyl receptor occupancy with a higher dose. This is just a quick look at the PK. We did also make sure, of course, that the plasma PK is in good agreement with the data. I'll go through that in a lot of detail. Okay, and now we get to some of the results. So in this case, we're going to start with a relatively low concentration of fentanyl, still high enough for an overdose, but relatively low compared to that 50 nanograms per mil that I showed you a minute ago that is generally observed in overdose situations. So when we have a 25 nanograms per mil concentration of fentanyl in plasma, that translates to about a 75% of the mu receptor occupancy by fentanyl. And in the yellow dotted line here, you see the fentanyl only trace. So basically, you know, this is just a time span of 10 minutes. And let me zoom in here. Time span of 10 minutes. And so the fentanyl is not really going anywhere anytime fast over those first 10 minutes. We're giving naloxone at time 0. And then what we're showing here is basically the effect of that naloxone in displacing the fentanyl from the mu receptor. And you can see that for this, you know, relatively modest amount of fentanyl in the bloodstream, the approved 2 milligram intramuscular dose of naloxone, and shown in the pink line here, does a pretty good job of displacing the fentanyl. And we've highlighted here the 50% mark. The 50% mu receptor occupancy is sort of a threshold that you need to cross in order to regain consciousness. So that was sort of our target in these simulations. So the 2 milligrams does a pretty good job, and you can see that it gets below that 50% threshold in about 3 minutes. Now, the higher concentrations, 5 and 10 milligrams, are, of course, doing a more thorough job of displacing the fentanyl, dropping the overall displacement down lower, and also get to below that 50% threshold sooner. So again, you know, we're seeing the trend here that the higher doses do a better job and faster job of displacing. But for this relatively modest amount of fentanyl in the bloodstream, the 2 milligram dose does a pretty good job. As you can imagine, it gets a bit more complicated when you get to the higher levels of fentanyl. So here we're showing you just what happens when you start at that 50 nanograms per mil. That's typically observed in overdose situations, which, as we showed you a few slides back, results in close to 100, I think it's 98% receptor occupancy of the mu receptor by fentanyl. And here the naloxone's having to work a bit harder to try to displace that fentanyl. And you can see that the 2 milligram dose IM, it gets close to that 50% threshold. And of course, there's variability among patients in receptor expression, et cetera. So it's quite likely that for some patients, we can cross below, you know, regain consciousness basically within the 10 minutes. But this is starting to kind of cut it close. And you may have to give another dose if you're starting with the 2 milligrams. The 5 milligram and 10 milligram, on the other hand, do a nice job of getting the occupancy below 50% in a much more timely fashion. And of course, in this scenario, time is absolutely of the essence because the patient's breathing has stopped. So the 5 milligram dose here gets below the threshold in about 4 minutes. And the 10 milligram does a little bit better still at 3 minutes. There's also a concern about what's called re-narcotization, where basically there's an upswing in the fentanyl, or rather in the, as the naloxone leaves the system, the fentanyl can actually regain some receptor occupancy. And in those cases, again, you might come down, come back up, and then need another dose sometime out from when you were first dosed. So again, you can see that the 2 milligram dose makes that more likely because you're just right around that threshold where consciousness may be lost again. Okay, so that's just at that 50 nanograms per mil fentanyl dose. Now what happens when you get to higher levels of fentanyl, such as what may be seen in more recent years? So here we're showing you again a range of different naloxone doses. Again, you can see that the receptor occupancy by fentanyl is close to 100% with that 100 nanograms per mil plasma level. And here the 2 milligrams just really doesn't get anywhere close to the 50% mark. You really have to go to at least that 5 milligram dose in order to displace enough fentanyl from the receptor in order to achieve efficacy resuscitation within the 10 minutes. 10 milligrams, which might be twice, 2 times the 5 milligram dose, would do an even better job of getting there sooner. Oops. There we go. Okay. And over here, we just extended that out to about 15 minutes, and you can see that the 4 milligram dose, which would be, the total dose if the 2 milligram dose were administered twice at time 0. That's still only just getting to that 50% mark at 15 minutes. So that's awfully slow to try to rescue somebody in an acute overdose situation. Okay, so what we concluded from the receptor occupancy modeling is if you just kind of anchor around the typical fentanyl overdose concentration of around 50 nanograms per mil, you're going to see a receptor occupancy of about 98%. And 10 minutes after naloxone administration at 2, 5, or 10 milligrams intramuscular, the receptor occupancy was estimated to be 50%, 29%, or 17% respectively. So in other words, that 2 milligram IM dose just gets to that 50% threshold to restore the patient's breathing at about the 10-minute mark. The timing here was really important as well. The time to drop below the 50% mark, given the different doses, was, again, 10 minutes for the 2 milligram naloxone dose, but only 4 minutes and 3 minutes respectively with the 5 and 10 milligram doses. We have demonstrated pretty conclusively, as you might expect, that higher concentrations of fentanyl do require higher doses of naloxone for effective reversal, where effective here is interpreted as a drop below 50%. And the lowering that fentanyl binding in the first, at most 15 minutes, by using higher doses of naloxone, we think should result in more rapid and successful reversal of the opioid toxicity and decrease mortality. So the overall conclusions from this project that we reported out to the client, and Jake will talk more about this in a moment. We do predict that higher naloxone doses do effectively reverse more opioid overdoses and should save lives. The simulations demonstrate the utility of the higher doses in displacing the fentanyl and doing it faster. Naloxone at 5 or 10 milligram reduce the fentanyl receptor occupancy below 50% for a longer time than the 2 milligram dose, so you have less chance of that rebound. And as you could see in the 100 nanograms per mil fentanyl simulations, naloxone, you really needed the 5 or 10 milligram dose to get that receptor occupancy below the 50% in a timely manner. Okay, so that wraps up the modeling portion. We do have a couple of publications about this model that give you a bit more detail about some of the modeling decisions and assumptions. And now we'd like to ask Jake to comment a little bit more about how the modeling was actually used to support the dose application.
Yeah, sure. So again, and I kind of alluded to this in some of my comments at the beginning of the presentation, but given the wide availability of the data, and as you kind of pointed out, we have decades and decades of published experience with naloxone. So being able to lean on that and to provide a model in addition to that, that really not only just supports a higher dose, but really speaks to what we're facing in the field, was something that was very attractive to the FDA and was resonating with them, and really helped to facilitate getting this out there. I mean, obviously the nature of the public health crisis itself, and current administration's focus, and previous administration for that matter, focus on getting our arms around this, if at all possible. We're also contributing factors. But again, as I look back on this and speak to the people that were directly involved, and even to people at the FDA and key opinion leaders in the field and so forth, this was a really interesting turn for the FDA to get a product approved so quickly, primarily on the heels of a model.
Thank you, Jake and Christina. And I would like to start. We have quite a few questions that have come up, and I'm going to start. I might answer some, but I'm going to direct others to Jake and to Christina. And the first question actually goes to Christina, and it is, did you ever measure the sensitivity of the model to the various choices of the binding coefficients?
Sorry. Yes, that's a great question. And the answer is yes, we did look at that. It really did not change our conclusions. So the conclusions were very robust to the possible variability in those binding coefficients. Of course, one could extend the model. We did focus on fentanyl here. As we mentioned, there are other synthetic opioids, many others that are out there. We focused on fentanyl. If you looked at a different compound, you would see certainly different PK, so different brain concentrations and different levels of binding. But I think the basic conclusions from this model are actually very robust to that type of variability, where we're not really just focusing on the absolute levels of the reduction, but also the key insight that the reversal will come faster when you go in with a higher dose to begin with, and that competitive binding. I think those are very portable assumptions across different compounds. But yeah, that's a very good question. But we're just showing you the one scenario here because it's really typical. It's also very consistent with what is observed in the field in terms of the multiple doses being needed to achieve resuscitation, et cetera.
Thank you. The second question is actually directed towards Jake, and it is, was the fact that the overdoses in the community may have been due to other synthetic opioids, even more potent than fentanyl, contribute to the agency's willingness to accept a higher dose of naloxone?
Yes, unquestionably. As Christina kind of alluded to, all fentanyl is not created equal. When we talk about these fentanyl analogs, and there was a paper published recently that suggested there could be well over 4,000 of these that were synthesized. I heard another figure recently that there was well over 10,000 that could theoretically be synthesized. And, these agents haven't been commercially available. And although we may assume that some of the kinetics and the dynamics of these agents may be similar, we just don't know. I firmly believe that they are different just based on what we're seeing in the field. And, at this point, the last number I saw was that the DEA had identified only 30 individual fentanyl analogs. And again, based on the evidence that we're hearing from the field, that just continues to roll in. And the number of nasal naloxone products that are required now to be administered to folks is just increasing. Every time I talk to a paramedic, do I talk to a first responder? It was when I first, started working with ZimHi about a year ago, it was maybe 2 to 3. I heard a story last week that there was 12 that was required to be given, and I'm hearing 5 or 6 regularly. So unquestionably, that was a big topic of discussion around this, as well as not just ZimHi, but Cloxata, which is an 8 milligram nasal spray, and other higher dose naloxone products that have been being discussed. But basically, the unknowns that are out there, but the clear need and the clear trend we're seeing with more nasal naloxone being administered was definitely a factor.
Thank you. So Christina, what assumptions do you think were the most influential on the simulation results?
Let me talk about what data were the most helpful, and then because I think the data that we're anchoring on, despite the fact that we had to make a lot of assumptions because of the variability in the field, the data that we really anchored on were, the plasma concentration data, where we had, quite reasonable amount of that, the data there. And then the receptor occupancy, given the plasma concentrations. That kind of allows you, given that we knew the affinities fairly well, that kind of allows you to infer the brain concentrations. So that was really important to kind of get that right. There are a lot of data around mu receptor expression in different parts of the brain. It can, gets quite complicated quite quickly, but I think we were actually able to find, really the key data to help anchor the analysis were pretty clear. So that was super helpful. And in terms of the assumptions, as I mentioned, I think you can, do sensitivity analysis, and one should, around the different assumptions for sure. And I can certainly, you could take different assumptions and get slightly different results in terms of when exactly you're crossing that 50% threshold, for example. But that's not so much the point as to just have that demonstration, the simulations that demonstrate quite clearly that, given all the available data, you get, that show that you are able to get to a lower level of fentanyl mu receptor occupancy or opioid mu receptor occupancy with a higher dose, and that time savings. I think that time savings was really a key insight, and that, of course, is anchored around the competitive binding and the relative concentration profiles of the different compounds.
Yeah, and if I can add to that real quick, Christine, I can tell you from a clinical standpoint from the field, that 50% threshold is what resonates. That's what's really catching clinicians' attention, saying, oh, I wasn't aware of that. Is really helping to elucidate the role of these higher dose naloxone products, and more importantly, an intramuscular product.
Right. Yeah. And one of the questions that has come up is the question about timing and the window of timing for naloxone intervention to avoid cardiac arrest if the patient stops breathing. I can mention that when we designed this model, the clinical team that we worked with asked for 10 minutes after naloxone is given. The assumption being that there may be a period of time before the naloxone is administered where the patient may not be breathing, but that after naloxone is administered, if the patient doesn't start breathing within a certain set time, the chances of survival drop dramatically. And Jake, you probably have spoken to people in the field about that.
Yeah, another very close second to the 50% threshold in terms of the weight of the statement in that discussion. You know, and if we use nasal naloxone products as an example, you know, the instructions are to give one, wait 2 to 3 minutes, administer the other, wait 2 to 3 minutes. You know, if you're a parent or a caregiver, that's a long time to wait. And even first responders are not waiting that period because we're looking for more of an immediate response. And again, I'm out there reinforcing that message that the longer these folks are their brains are deprived of oxygen, the clear risk of not only just death of brain cells and damage to brain cells, but cardiac arrest is imminent. And again, that's just something that, believe it or not, is just starting to resonate with a lot of people in the field. I think, again, clinicians understand it, but, you know, explaining that to first responders especially is, you know, you sit there and you see the light bulbs start to click and say, you know, yeah, I would rather have this product than what I'm currently carrying.
Thanks. So, Christina, there's a couple more questions, and several of them ask about some of the variability. So inter-individual variability and looking at variability within the platform. So I'm going to summarize those as asking, did you look at what effects variability might have in terms of the platform and the response?
Yes, and I think that's similar to the question around sensitivity analysis. So, you know, we showed you, like I said, the sort of best guess, middle of the road predictions. Certainly, you know, as we mentioned, different users could have different sensitivities to the opioid, different levels of receptor expressions, somewhat, you know, different recycling dynamics, etc. So yes, we did look at that. We, you know, could show you almost infinite number of, you know, other scenarios that look a little bit different, but they would all share kind of the same basic conclusion, which is that, you know, you need more naloxone, and time is of the essence. And like I said, the time courses that we got out of our simulations were very consistent with what's observed in the field. So I think it's probably representative of an average scenario. Are there other scenarios? Yes, but would you do anything differently because, you know, the patient might be a slightly different patient? I don't think so. I mean, you know, as Jake just explained, time is of the essence. So having that higher dose product available is going to be beneficial regardless of the variability.
So I'm going to take the next question. It's actually several questions asking about the could the model be used to assess the impact of the mu receptor internalization and recycling on receptor occupancy? And yes, it could. And in fact, I think Christina also mentioned in discussing the model, we had to assume that the person receiving the fentanyl was a chronic opioid user. And we did this because that person would have a different internalization and recycling of the receptor than somebody who was opioid naive. And that's a very important factor because people who are opioid naive tend to require less fentanyl to have an overdose. So we were specifically targeting part of the population who might have a desensitized receptor with a change in dynamics so that they would be requiring and taking larger amounts of fentanyl. So we have another question for Jake, which is, can you talk a bit about how the modeling results are currently used?
Yeah.
Or are they
So,
continuing to be used?
yeah, it's a big part of our clinical presentation. To be honest, I mean, addiction medicine is a very unique field, and I've worked in many areas of medicine. And what I mean by that is there are very strong beliefs, whether they're, religiously based, theoretically based, just, strong feelings around, a lot of people in recovery working in the field. And we have a lot of folks that give us pushback saying it's too much naloxone and you're going to precipitate too much of a withdrawal. And, there's some other sporadic clinical concerns. There's been some potential evidence of pulmonary edema, or suggestions, I would say, as opposed to evidence, because it's very, very thin. And, basically pushing back on the need for these higher dose products, and, being able to utilize this modeling as a primary tool from a clinical perspective, whether I'm presenting to health plans, whether I'm presenting to clinicians, again, even first responders, to explain this as in a less technical term, but just the idea of the faster we can get the fentanyl off of the receptor, the more likely we are to wake somebody up, is very, very valuable. So I'm not, exaggerating here. This is a primary tool in my tool belt that I'm using to educate folks, throughout healthcare. So without it, it would be very challenging. I would be up against just people's beliefs and opinions, and not have a lot of clinical evidence to support it, other than saying, well, we think we need higher dose naloxone because of these fentanyl analogs. Now, let me have a model that I can show you that explains why these are important. So it's been essential. It's been very, very helpful. We wouldn't have gained the traction that we've been able to without it.
Thank you. Christina, I have another question for you. And we actually have several people with a similar question, which is, did you look at administration of fentanyl with other drugs that perhaps inhibit or change transport or metabolism?
Yeah, that's a very good question. And we have not done that. That might be an interesting extension to this work. Yeah. It's a good idea.
I think we've addressed a number of the other questions about safety of naloxone and use of higher doses. I did have one final question for Jake, which is, where do you see naloxone and its use going in the future?
Yeah, so I mean, it's clear, and everybody knows this. I'm not telling anybody anything that they're unaware of, but we still have a ways to go in naloxone distribution. We have a lot of it out there. We don't have enough of it out there. There's been models that have been done, looking at states' ability to saturate basically their naloxone needs, and out of their 50 states, only one, and that was Arizona, met that model's criteria. So there's a lot more room in general for naloxone, and I firmly believe, although it's been somewhat slow and challenging, just again based on what we're up against and people's beliefs about the potential pitfalls that exist of administering too much naloxone. Just in the 13 years that I've been working in this field, just seeing how these trends go, we're getting to a point where it's very hard to argue against higher naloxone dosed products. In addition to that, based on some other recent data that's been presented, the route of administration makes just as much of a difference. An IV and intramuscular really seem to be the preferred route in terms of the ability to prevent cardiac arrest. So I think in the next year or two, we're going to see some dramatic shifts towards the acceptance of that. and these will become a standard of care.
Thank you. I'd like to thank all of our attendees for the many, many questions that we received. And for those who've asked about the model and the modeling, I'll point you to the publications, particularly the one in PLOS 1 that are available. And with that, I'd like to give my huge thanks to Jake and Christina, and to all of our audience for a really very informative and very nice webinar. Thank you both.
Thank you.
Thank you.
Have a great
Rebecca.
day, everybody.
You too.
Bye.
Thank you. Bye-bye.